Hey, everybody. So I had the pleasure last night to go to dinner with Patricia, but and maybe we drank a little too much wine, but we had some fun. And Patricia's a little nervous today for some reason. I don't know why, just because the YouTube's going to happen, and there's all sorts of fun things that are going to happen today. And then I'm supposed to sit and make faces. But I thought it would be neat if we had a introduction for Patricia that was a little more in-depth than what I could give. So I asked John Towns to come up and give an introduction and then actually maybe throw a story or two out before Patricia started.
Is the YouTube started yet or not?
I hope so.
A flashback from 20 years ago.
Oh my God.
So I'll do the obligatory about but I read what was put together for her background, and it ends with her last position, which says, well, she was a director of an NSF supercomputing center, which, okay. But that turns out that supercomputer center was NICS at University of Tennessee in Oak Ridge, which is a partner in TeraGrid and Exceed and all these sorts of things. So I've actually known Patricia for 20 years. First time I met her, she didn't think I liked her. But, so let me do the obligatory. So she's currently at Mount Sinai, as is said here, Senior Associate Dean for Scientific Computing and Data Science. And she's running a bunch of NIH projects at this point in time. Most of this I know less about because I've kind of lost track in the last couple of years, other than I see your name pop up occasionally on various fronts. So the Children's Health Experience, Health Exposure Analysis resource, and you've got a couple of other data-oriented projects, Community Research, Education, and Engagement for Data Science project. That's another NIH project. And I expect you're going to talk a bit more about those sorts of things in this, so I'm not going to say too much about those. But she's got a lot longer history in this community. Again, the first time I ran across Patricia she was in Albuquerque. And oddly enough, the first time I think I met her was in a former Cadillac dealership building.
Oh. So
at the time, University of New Mexico was a partner in the Alliance, for those that go back that far. And they were running some systems, and the center at University of New Mexico was established in a former dealership.
including the fountains and everything.
And the large glass windows,
Yes,
which is really
yes.
great for cooling control in the
Yes,
desert
yes.
and all these sorts of things. And so, a bit more of her history is she went from there to San Diego, another partner we know a lot about. And from there, there wasn't anything between there and Tennessee. So she was then at Tennessee and became the director of NICS before she moved on to Mount Sinai. So she's got a lot longer history with this community, and many of us know her better than might otherwise be obvious from the introduction here. But so I'm very glad to see Patricia here, and I'm interested to hear what she's been up to in the last little while, because I have lost track a little bit. But she's made a lot of great contributions to this community over the years, and it's great to see her having made this step in this direction, because it's a little bit outside our community in some ways, and to establish the use of these sorts of resources and services in the context of medicine, which is still a burgeoning area. And I want to hear about the cool things you're doing.
Okay, thank you.
Welcome.
Thank you for that. Thank you. Yeah, I was actually thinking about this, that the first time so first of all, I want to say it's a total pleasure to be here yesterday. Last 2 days have been really super fun and super interesting. And yeah, as I was coming here and thinking about this, I was thinking about when was the first time I worked with NCSA, and it was when I was in New Mexico, and it was with Mike Showerman and Rob Pennington doing a Windows cluster versus Linux cluster. And so we all know how that turned out. So in any case, so yeah, it's been 20 years. So it's really a pleasure, and especially meaningful for me to be here because I've gotten a chance to work with all the NCSA folks for so long. So I just thought I'd start a little bit about Mount Sinai. It's a private medical school and hospital system in New York City. We have over 10 million patients. You can see we have like 18,000 babies that are born each year. It's like a small village or whatever, and about almost 4,000 hospital beds. So it gives you some sense of the scale and scope of the Mount Sinai Health System. In this picture, you can see, so we're kind of in this like 4-block radius here. On the upper, so you're on the upper, we're right between the Upper East Side and East Harlem, and so you're kind of looking over the park. Let's see, Columbus Circle is on the other side, like where you can almost see that CNN sign. So just to give you some idea of where we are. But of course we have locations throughout the city, but we're one of the largest employers in New York City, about $7 billion a year. So it's, I think we're one of the largest health systems in the country, and in the last couple of years, we also got an NCI Cancer Center designation, and we're working towards getting a comprehensive cancer center designation. So it's an exciting place to be with lots of stuff going on. And again, I don't know how well you can see this, but this is just showing you kind of some of the locations that we have throughout the boroughs. With mostly in Manhattan, we have lots of clinics and hospitals, but then also we have partnerships in Westchester and other places, Long Island, in New York. And I just thought, having lived in other parts of the U.S. And knowing what it takes to build a building and how much it costs, I thought it might be interesting. So this is from 2013, but this is a new building that we built, and the first 7 floors are all clinical space, and then we built this residential tower on top of it. So actually some of this 33 floor residential tower, so actually some of the income from that helps to pay for some of the operations of the clinical space. So, New York City has interesting challenges, but we also have a computer room in the basement of this building too, and we're just beginning construction on a new building that's going to be like a block up from this too. So, but of course in New York City everything takes longer to do than you would in anywhere else, and it costs a lot more. I'm always like, what do you get for like $700 million in New York City? And this is what you get. So, or at least this is what you got back then. So, okay, I wanted to talk a little bit about some of the science drivers that kind of help drive and drive the things that we do. I came to Mount Sinai, I guess it was 6 years ago now, from NICS, as you said, and so I was recruited to help build up a scientific computing infrastructure, and I also oversee the Mount Sinai data warehouse, so oversee all the data warehouse for all the patient medical records and a lot of the electronic data capture systems for clinical trials. So it's so I'll go over this, but so of course having the science drivers and understanding what the clinicians and researchers are doing and feeding that into being good partners and providing a good infrastructure to make the whole system work effectively is obviously very important to me. So I'm going to talk a little bit about what we've done at Sinai, some of the impacts of those efforts, and then also some of the outreach and bridging communities and some things on education and training. So as far as precision medicine goes, I think a lot of institutions and people have a similar view of this. I mean, in some sense, it's taking all the information from the digital universe and using that to build a predictive model of disease that's specific for a person. Because, 2 people will have cancer, you give them the same drug, it works for one, it doesn't work for other. Why is that? And what can we do to improve the diagnosis and treatment? So, how can we use all of this information to do better at this? So, of course, there are some therapies and immunotherapies and other things where there have been effective treatments, but obviously it's still not effective for everyone. And I think, right now you try to figure out what the driver mutation is, and then you can treat it with a drug focused on that. So this is something that we do at Mount Sinai, where you sequence the tumors, you try to understand what the mutations are, and then we do the molecular dynamics simulations to help figure out which drugs would be targeting and to work on that. And sometimes it's effective, sometimes not. So not all cancer patients derive clinical benefit from this mutation-based therapy. So the question is, why? Obviously there's lots of things here that we just don't understand. And so as we work towards to try to understand this, I mean, part of it is that the cancer cells don't operate in isolation, but a lot of our data sets that only have one type of data in it. And so how can so obviously we need to be able to combine different kinds of data and multi-scale levels of data together to try to understand what's going on. I mean, as you can see, here's some of the key questions. What components are present? How are they responding? When does it happen? What causes a tumor to go metastatic? What are all these kinds of questions? So one of the, to me, one of the most exciting things that has just been announced just a couple weeks ago from NCI is, getting a little ahead of myself here, but is that NCI wants to build a Google Earth for tumors, right? So how can we visualize and represent all of this multi-scale data from the molecular, the whole tumor, the tissue? How do we represent this in a 3D way so that we can try to see what some of the interactions are that are happening and to see it over time, right? So some of it's just trying to better understand this. And so this is really important. And so NCI has just announced this new Human Tumor Atlas network. And so this is to replace the Cancer Genome Atlas, TCGA, which some of you may know about. So this is the follow-on for that. And so NIH has announced there's going to be these research centers. I think they're going to award 3 of them that are kind of focused on specific tumor areas. And then there's going to be there's going to be one data coordinating center. So all of the data that's going to be collected from the research centers gets, put together into the data coordinating center. And then of course it's going to be a resource for the whole national community, and it will work with the other data sources that are in the overall cancer ecosystem. So this is obviously to me, this very important part of this. I'm sorry, but I'm going to actually just show you this slide now because this is part of it. So one of the things that NCI has been funding, and this has come out of some of the Blue Panel recommendations, is there's a genomic data commons that's already in place, and that's being run by Bob Grossman at University of Chicago. They're also starting up an imaging data commons. There's a proteomics data commons that's going to be announced any day now. And so they're trying to build these resources so that to have the greatest access to data is possible for the broader research community. So this is just another piece in that puzzle. And actually, and I'll talk about, and there's other ones that are part of this. So in any case, all of this efforts is part of the Cancer Moonshot, which I think everyone probably heard about. And there's been a lot of money committed to these projects. So some of these announcements are coming out as part of this. Perhaps you've heard of there was a DOE and NCI, some pilot projects that were announced last year. And again, it's like developing some new computational approaches. The first one on here is around molecular dynamics for RAS-related cancers. I mean, there's other projects that have machine learning, looking at medical records, lots of things. So there's lots of focus, and NCI is looking to other communities to help leverage the expertise that they have. So to me, this all makes this a very exciting place to be. I pulled out from the Blue Ribbon panel some of the recommendations that were informatics, data science related. And so, one of the things that I've learned is that recruiting patients for clinical trials is very difficult. And so some of it is around, I think people don't know if they qualify or what's available. Maybe some people just don't want to participate, but there's a lot of effort around helping to identify patients for clinical trials. And I think some of it too, as you can see in this first one, is getting patients to help contribute. You can go to something like Patients Like Me. I don't know if you've ever heard of this. Especially in rare diseases, there's a lot of websites and communities that have developed where people readily share their medical information as they try to understand more about what's happening and why, and share their experiences. So in some sense, this is trying to leverage a lot of that community and that kind of experience. I mean, of course, number 8 around, again, this is clearly like more of a machine learning thing, trying to do this predictive analysis, building this national cancer ecosystem, which I've been talking about, again, making more and more data available for people to mine, and developing this cancer atlas. So, I think, the recognition is that to go forward, it's this availability of data is really important to try to really understand what's going on. So this, unfortunately, you can't see the white bits from the panda so much, but I mean, to move into this area of data sharing, the panda says, will you share your data? And the bear, I guess, says, everything you need to know is in the article. And it's like, no, what I need is the data, right? And I think part of this is that trying to change the culture around data sharing and with the communities to make the data more open and available. And also, some of it's for research reproducibility, but some of it's just to better understand. If you have more data, you get greater statistical power. You can understand more about what's happening. So that's helping to drive a lot of these efforts around the data commons and the different data sharings from NCI and NIH. And again, there's an office, I'm sure a lot of you here know Phil Bourne, who did the Protein Data Bank and was at UCSD. He was running the Office of Data Science at NIH, and he helped to develop these FAIR metrics, in making data findable, accessible, attributable, thank you, interoperable and reusable, and somehow giving recognition to all the people who have created the data. I mean, you've probably heard Phil Bourne say, everyone quotes my the Protein Data Bank paper, but no one's read it, right? And so how do we give, and especially for the folks here who are who I expect are often very involved in the creation of data, and so how do you give credit to the people who are really part of it? I mean, you think about LSST and the other physics communities where you'll have like 100 authors on a paper, but people are getting credit for, for collaborating, and I think in the biomedical space this is more new, and there's trying to figure out, even for tenure, for faculty and all these things, how do we give credit for people who are being the creators, the collectors, curators, and managing all of this data, and not just coming out with the papers on it. So anyway, so this is a big topic of discussion. One of the other things, too, that is perhaps of interest is this was from a JAMIA article, and you can see how there's all these different data types, the structured data, unstructured data. So a lot of what we're trying to do is find some way to be able to derive meaning from all of this, right? And it's, and you can see a lot of, like for instance, some of it's, I was telling some of these stories already, but you think about you go see the doctor, and instead of writing, instead of clicking the button in the medical record that this person's a smoker, often that gets put in the physician notes, right? So then someone does a study on all the medical records, and they want to do a study on smokers who are on this drug and this other drug, but of course they miss a lot of the smokers because the smokers, because the physician never clicked the button for smokers in the medical record. So there ends up being, we can, so there's lots of data there, and some of it's trying to figure out what's the quality of it, and I'll talk about this a little bit more later. But as you can see, there's lots of different kinds of data, and so how do we correlate all of this with all of the data that's out there, and how do we derive some kind of meaning from it? Also, you probably heard about the Precision Medicine Initiative that's now called All of Us. And so, of course, as a computer person, I start thinking about this, especially, how large is this data going to be, where, especially as part of a lot of the exascale, is, when you think about exascale and where are we going to need to be and what kind of technologies and applications and things that we're going to need. So here's just a back of the envelope calculation about what it would take, for the 1 million people that are going to be recruited and are going to be sequenced as part of this initiative. Just the raw data, this is without any subsequent processing or anything, we're already at 300. We're already at like 400 petabytes of data. Then, of course, we're going to want to integrate it with all those other data sets and correlate it. And so, as you can see, obviously, this ends up being a large compute and data problem. And so So I'll say, so, I mean, of course, and there's other kinds of things. So images, of course, are also important, also need to be correlated in part of this. So like one of the, I just wanted to go over this. So another driver for a lot of the computation and data infrastructure. So you can see on here, I guess I'll say the depressing, or maybe I'll say, I don't know, what's the right word? I guess I'll just stick with depressing. So you can have, an experienced radiologist reads an image correctly about, 65% of the time. Two experienced radiologists gets it up to 70. And I've seen a couple different studies. Sometimes they're a little higher numbers, a little lower. But I mean, even with computer-aided diagnosis software and doing all the image recognition and to the best that we know how, it is more helpful. Can get it. I've seen even studies where it's in the 80s, but it's still like 80% of the time. Like, this is clearly not enough, right? This is clearly not enough, right? So, and I think even in like people, one of the radiologists that I'm working with, even when she's, the technician is doing the ultrasounds, and it's highly dependent on who the technician is. So sometimes the technician will just miss something, right? Or they'll get something and they don't, like, they'll either miss something and then the radiologist, they're looking for every ultrasounds that they're getting. It's like 3,000 to 5,000 images at a time. So, there's fatigue, there's lots of reasons why things are missed. And so again, what can we do to help with all this? So, and you can see on this slide, of course, from the radiologist that I'm working with, the main thing is, the stress from the false positives. Missing stuff is terrible, but also, letting people think that something is wrong when it's not is terrible. And, the percentages you can read on here, I mean, all of this is way higher than it should be. And I don't know that this is unique specifically. This happens to be for breast cancer. The last one was on lung nodules for lung cancer, but I think, it's similar across all the domains, is my understanding. So you can see there's real drivers here. And also, again, thinking about, for this all of us, where the 1 million patients are being recruited and getting all the genomic analysis done, this is, if you think like you have 20 images, already you're at an exabyte for 1 million patients, right? So how can we do good feature extraction on all these patients? The images are all different, shades of gray and all these different things. So how can we do this in a consistent way that we can really have software that really helps the radiologists do this better? I thought everyone here understands molecular dynamic simulations. Of course, this is also an important part of precision medicine and also a driver. So I wanted to include that. Another thing that to me is also part of all this is being able to do multiscale modeling on things like the heart, right? So again, being able to understand enough about how the molecular interactions and the cells and the whole organ is interacting with the vascular system, and being able to do it in a way that you can do it in less than real time, right? So, I mean, right now, even the best electrocardiac models, it's like doing a heartbeat takes longer than a heartbeat actually happens, right? And obviously you want to be able to do this and make it patient specific, so you could say someone's going to have a heart attack or something's going to happen, right? And then ultimately you want to, interject different drugs and all these kinds of things to help predict what's going to happen and help to hopefully influence whether it's behavior or intervene with drugs or whatever it is. So again, we're obviously very far away from this, but to me, this is the long-term vision of what it would be, what we want healthcare to do, right? And so, and another project I just wanted to mention that is also that I've gotten to sit in on a neurosurgery simulation, which I told some of the folks yesterday. It's like the most amazing thing I've ever seen and the most horrible thing I've ever seen at the same time. Neurosurgery residents, it takes 7 years for the whole residency, and then you're not allowed to operate on anyone until you're in your 5th year. So what if you're not good as a surgeon, right? So some of this is having really good tools. You have flight simulators, you have all these other kinds of simulators. we need tools to help identify, who's going to be good at surgeons, but then also we want to be able to practice the surgery in advance. If we could practice it, then you could see that this is in the way or that's in the way, or if we could get good models with the bleeding and whatever, you can help prevent things and spend less time in the operating room, obviously, which saves everyone money and stress and everything. So there's obviously really good drivers here for why this would be important. To me, it's like I want to give everyone a 3D printed tumor, right? And help people understand how it fits in and what's going to happen, because I think, you want to know what's happening, right? And really understand it. So, so obviously, there's all kinds of things here. And some of the things here are things that we know from like the blood flow is like a lot of the turbulence simulations, right? There's things that we can take from other fields and bring to bear to help do this modeling, but again, it's we still have a ways to go. Okay. So some of the things that we're doing at Sinai, I thought this might help to give an idea of how, how we are, some of the expertise that we have and some of the efforts that we have and how it kind of fits in the overall scheme of things. So you can see on top there's the different research targets, the molecular and cellular processes, the tissues and organs, patients, and then of course population and society. And then there's this American Medical Informatics Association that they have their 5 defined specialties in bioinformatics, imaging informatics, consumer health informatics, clinical informatics, and public health. And so you can see some of the different projects and some of the different institutes and all these things that kind of that we are using to help solve clinical and research problems in these areas. And then, of course, there's all these different approaches that we could use and different resources that are available both at Sinai but also nationally. So it's like trying to help share some of the things that we're doing and how it's organized. And so I would say our overarching aims is to make data more accessible, to the researchers and clinicians so it can be used. That it means a whole variety of things, right? Like working with our local institutional review board to help them understand how we can, so they can understand our different processes for security and for making it available, and helping to come up with different processes for researchers too, so that we're helping them do the right thing. So there's things like you can give out de-identified data, but you have to agree to not re-identify it. Every institution kind of interprets the privacy issues in different ways, and so having good relationships with all of these groups helps to come up with I'd say technology solutions and processes to help make these things more available and useful. And then also we want to make the data more actionable, right? So I think some of this is around some of the reproducibility things and some of the other things, so that the data is more useful and you're serving it up in a format that people can do something with it. And so, again, I try to organize these in some ways that can help show some of the different areas, some of the different efforts and how they fit into these different areas. So, we have, EPIC is the medical records that we have, medical record system, so they have their own kind of mobile apps and other kinds of ways to get in and interact with that. And so we've developed some other interfaces and ways to interact with that and be able to communicate information, and also help recruit people for clinical trials and things like that. We've spent a bunch of time on doing e-consenting, right? So you go to the doctor, you want to give everybody an iPad so that they can consent for trials. And in the past, usually it's been, someone has to sit there and explain everything to you, and now you can do it in other ways, and it can obviously is more scalable from a patient recruitment standpoint. But there's lots of other efforts, like we've been funded to build this research imaging data warehouse. So we have over 1 petabyte of images in many different modalities, and we're stripping out all the personally identifiable information in it and then making it available on our local supercomputers, so people can do feature extraction and try to see if they could see the tumors sooner, or they can compare different patients, different sorts of patients together. So you could, in this case, what you would do is you would go to our data warehouse. We have a cohort query tool. You could search on certain patients with certain kind of characteristics and find the ones that we have images for, and we act as the honest broker. So you could do your feature extraction, but then you want to know more about certain patients, you can come back to us, we can re-identify them for you, and all of those sorts of things. These electronic data capture systems are used for a lot of clinical trials. They collect survey data. We have them connected to our medical records so we can pull out data directly from the medical records. There's all the tissue banking stuff. We have a biobank that has serum on 50,000 people. We're about to receive exomes on all of those people, and then we have all of the, of course, all the medical record data that fits with that. So that's another rich data source that we want to be able to leverage and use. Let's see. Some of these I'm going to talk about later. So, but you can see there's many projects, some national, some local, regional, and of course the training's really important. So I'm going to talk about that later. And you may have seen Sinai's asthma app. I don't know. That was one of those in the Apple Store. So it was like, 25,000 people downloaded it in the first day and signed up for all the trial and all this kind of stuff. So there's a lot more patient engagement through apps and things like that. So. And actually, maybe I'll tell a quick story about, there was one, I was working with a psychiatrist who, you have they have apps, you can put your feelings and other things in the apps, and he was doing a study on OCD people, and, I was thinking, I wouldn't put this information in an app, but he was saying, no, with OCD people, if you tell them to do something, they do it. I'm thinking, anyway. Okay, and then I just wanted to share with you some of the components, because, to make all of this, maybe I'll just say to make precision medicine happen, but really it's perhaps even broader than that. You kind of have to have all of these different systems in place from a technology standpoint. I was talking a little bit about the cohort discovery, and being able to identify, the first thing a lot of researchers want to do is, how many patients are there like this? How many white males over 50 that are smokers that are on this drug and this other drug? Because they want to see, were they also cured for depression? Because there was some weird drug-drug interaction that helped that to happen. And so the cohort discovery is really important, because that's almost like the first thing people want to do when they want to study something. They got to know, how many people do we have like this? And as more data becomes available nationally, especially for rare diseases and other diseases where there's just not a lot of patients in that area, being able to do this and have much larger and richer data sets is really important. I'll point out, I don't know. I guess maybe I talked about some of these things already, so maybe I won't necessarily point out so many of these things already. I mean, the limbs are really important for tracking all these samples and all the tissue management. I mean, there's a lot of software and it's almost like these are all almost separate pieces of software and infrastructure. So I think one thing that's really important is connecting these together in an automatic and reproducible way, so that it is easier to, whatever it is, whether it's a clinical trial and we're trying to process tissues in a certain way and be able to, and also for regulatory reasons, we want to be able to show that everything's connected and looked after in the proper ways. So it's also making all of this software connected and is important. So I talked a little bit about these electronic data capture systems. So we actually have a couple different these are systems that you use for surveys and things like this. Someone enters a clinical trial and you want to capture all kind of data that they give you and also data that you have about them, and then you want to be able to compare it and do some discovery on the overall things. But I'll just point this out, and you'll see this in other later slides. I mean, even so many projects are collaborative and span multiple institutions. So even these databases that we have, I mean, they're used by countries all over the world because patients get recruited from all over the world for these different trials. So we also do to do all the data management for we have a grant, or I guess it's we work with NIOSH. It's not a grant. It's collaborative agreement with NIOSH, and we are tracking all the 35,000 9/11 first responders from the World Trade Center. So there's 7 different clinics in New York they can go to, but then there's one data center that collects all this data together, and obviously that's also a rich source of data that lots of researchers want to access and try to understand what happened and what were the effects of a lot of the environmental things that happened then. So. Also, as I mentioned before, I also oversee the data warehouse where we take all the medical records and we have all of these different data points. We have some, I would say, basic interaction where we can give physicians some genomic alerts and some other alerts based on genomic information through the medical records. We're working on building like a genotypic, phenotypic data warehouse so that we can, again, be able to correlate and give researchers and clinicians more access to a richer data set. And of course, we're working with some different local and national projects to share de-identified and some aggregated statistics, again, for either patient recruitment or for research purposes. So there's lots more to be done here, but it gives you some idea of the scope of what we have and some of the things that we're doing. I talked about the radiology exams a little bit, and I also thought you might be interested in some of the numbers on what we have. And again, this is all about the feature extraction and really trying to derive meaning from what's there and to be able to consistently recognize things. There was a project that we've been working on with an applied mathematician at Stony Brook where like tumors that have a fractal-like pattern on the outside are malignant, but the ones that don't aren't. And trying to see how to consistently recognize some of those signatures so that, again, we can come up with some better clinical decision support, and some of that stuff is really exciting and important. Let's see. Another project that we're working on is kind of building a data science pipeline, including one that's like a streaming one. So the example I have here is around this modified early warning system. So there's I don't know how many, 5 features that are part of the standard Muse score that the goal is to kind of predict if a patient is going to get worse, like go into sepsis or something happens. So this would be someone who's in the ICU or something like that. So the first thing we did was we did a retrospective study of all these patients, and we know what their outcome was. And so you try to see what are the physiological features that they were exhibiting before they got worse, and you try to figure out which ones are important and which ones aren't, which ones may be predictive. So we came up with a list of like 100 different features, which of course are weighted in different ways, but that become predictive of when someone is going to get worse. So we are now taking that, and we want to now make this streaming and do this in real time so that we can actually tell the physicians or nurses or whoever's that someone has just all of these, 100 things have gotten worse, and so please take some action because especially right now, there's a big effort around it. It's called failure to rescue. So if you know someone's going to get worse, you really you want to do something about it. You want to rescue and anything that you can. You want to do the best you can at caring for people. So we're going to be getting very close to using this in real time and trying it out. But then the infrastructure that we're building that others will be able to come in and use later and be able to plug in their own machine learning algorithms for other projects. So this would be, other projects would go to the IRB, the Institutional Review Board, and they would say, we have access to this data, to these different data types. And since we have this pipeline connected to our data warehouse and these other sources, then we can help make that data available. People can plug in their own machine learning and they can iterate as well. So it's putting in new infrastructure in place and making the data and everything more available and easier to use for research. Okay, As you know, as a supercomputing person, this is a picture of our mini supercomputer that we have. It's about 13,000 cores. We have 160 terabytes of flash. We have about 20 terabytes of GPFS. And we're using TSM for archival storage, and people have actively been using that. About 2/3 of this gets used for genetics and genomics workflows. 1/3 of it gets used for molecular dynamic simulations. And again, so to me, one of the interesting things is how many outside users we have on the machine, right? And again, this is because there's so many collaborative projects that in some sense we're operating, our own little mini NIH computing center, I guess. And you could see, so all these different institutions. Hold on. So with this large genomic workload, I thought, of course, if the computing is kind of matched to the science that you're doing, you can get more science done. And so one of the first things we did when I first came to Sinai, we kind of built a small computing system, because, maybe I should apologize in advance to all the application folks in the room. But whenever you ask users, what do you need or what are you going to do, that you get a whole bunch of answers. But I mean, to me, it's always good to look at the actual data and see how people are actually using the machine. And it's not to say things aren't going to change over time, but you look at the actual data and see. So one of the things that was very surprising for me and interesting was learning about this GATK pipeline, which, like I said, this accounts for a lot of the use on the machine. So we have a genomic sequencing facility that's about $100 million a year genomic sequencing facility. We've come up with a bunch of different genetic tests that we discovered in different genes and stuff for some specific diseases. And in fact, this genetic testing facility, we've now spun it out as a separate company. But all of that sequencing and processing that goes along with that gets run on our mini supercomputer. So in any case, for those of you who are not familiar with GATK, a typical job is going to submit 10 to 20,000 jobs at once, and these jobs are all very short-running jobs, right? And then you have all the anxious grad students and other folks who want to know when their job's going to be done, right? So they write other scripts that are monitoring all of these. And some of it is in, how GATK works too, because it's looking to see when the jobs die or finish, and then it submits other jobs. So it's an interesting workflow that I was not expecting. This shows you a little bit about what some of this workflow actually looks like. And this is from 2013, but you can see, when I first came, I was thinking, one node, one job. I mean, that's how you do it in supercomputing land, right? You want that job to take advantage of all the memory and all the I/O and all of the capabilities of that node. But this workflow is very different. And so we actually share nodes and other things that, again, I was not anticipating until I saw what the workflow actually looked like. 62% of jobs are using only one core, and you can see how many jobs. This was only in half the year. There are 4.5 million jobs run in half the year. So it's just a different profile than what I was expecting. The other thing that was interesting was the I/O, and this to me was almost the more interesting part of it. So with GATK, you'll create a 600 gigabyte directory that has 1 million files in it and 10,000 folders. Many of these folders are empty, and you can imagine what this does to the metadata server for the file system. It's just a whole different kind of approach to computing. And so it's, how can we, so I want to create a system that's going to work consistently, perform consistently well for these kinds of workflows. This shows you a little bit of the distribution of the file sizes. So this was in 2013 on a 1.5 petabyte GPFS, but you can see like we had 200 million files, half of them were less than a kilobyte. And then like all the file, you can read the statistics, but it was just very different than a normal or usual supercomputing workflow. And so we did a bunch of things to help make these jobs perform consistently well. We were, with GPFS, you're able to create 4K inodes, so we were able to store a lot of these files directly in the inode, so they never made it to disk. That's also why we have 160 terabytes of flash, because then all the files that are a little bigger, we put those right on flash, and they're never going to disk. So, and of course we did benchmarking what was using most of the compute time. We did a whole study to figure out what jobs were taking the most compute time and finding the right processors to match with that and changing out the schedule. So we did a bunch of effort, and we have a paper about this, and I'm happy to talk about this in more detail, but it's just, changing, unfortunately, we can't change community codes necessarily so easily, and so it's coming up with other ways to have good, consistent performance, and people can get more science done. That's our goal. So, okay. So now I'm going to talk a little bit about some of the projects that, we spent, like I said, we spent a lot of time on this, on building up this infrastructure and partnering with different researchers and clinicians across campus so that we could have this effective infrastructure. So these are some of the fruits of our labors. So one of them is that we received from the National Institute for Environmental Health Sciences this award where we're the National Data Center for studies that have been done with children with environmental exposures. And so a lot of these, and I'll just say this was an interesting proposal because we have an epidemiologist, we have a statistician, we have a computer scientist, we have different people on complementary expertise on the proposal that helped make this happen. So there's a lab, so anyone who has some NIH funding can ask for their samples to be processed by the overarching CHEER lab network, and then all of that data comes to the data center. So we've done some things where most of the environmental studies are small, they're small groups of cohorts, and so we have a whole ontology and other things that we are using to help try to tag the data in certain ways so we can combine data from different studies together, again, for greater statistical power. So this has been a really interesting project to really try to understand and help make it easier for environmental exposures to be included in other studies. So, and also, I'll say, this is something that is also very important about this. So I think we were talking about earlier about some of the tumors and how much of the data sets that are existing right now are only, about the tissues or about, the molecular structure and things like that. So it's the same thing here, but in a different way where, depending on when you get exposed to something in time, there's these windows of susceptibility, especially as a child. And when you get exposed to things at certain points in time, it can have different outcomes. You can get exposed to the same thing but at different times, and something happens one time and something doesn't happen. But also the interaction of the different environmental exposures and chemicals also interact with each other. So often it's just a lot of the existing studies are just looking at a certain chemical discretely, but there's studies obviously that show that the different chemicals interact with each other and provide different kinds of effects. So this is something that is one of the main goals of this center, is to help enable these kinds of studies that are more accurate to how things are in real life. And so again, a very interesting project. We also there's this new Cancer Immune Monitoring and Analysis Center. Again, why do some patients respond and some don't? This is around all the immunotherapy stuff. There were 4 centers research centers that were awarded. You can see them at the bottom here. And then there's a data center that Dana-Farber got. So again, this is like a similar proposal where there's the different research centers, and then we connect all of the data, and the data ends up being part of the overarching data commons that I talked about earlier. But again, these centers are all around having this multidisciplinary expertise, people in different areas of microbiome, genetics, all the immune profiling and all the pathology, it takes a whole village to bring to bear on these projects and people with different expertise. And also, since I'm in the Genomics Department, I figured I would also include this. So there's been, as part of precision medicine effort, there was a massive effort at Sinai, put a lot of funding and effort put into our Genomics Department. So we now have about 100 faculty. This was also, in some sense, part of my recruit to help provide all of the infrastructure and other things to help enable all of this research. And so as you can see, we've changed our ranking in a very short period of time from 31 to 4th in the country. So we're obviously very proud of this, and our effect on being able to make some progress in this area from a scientific standpoint. Okay, and then this is my last section, so I just want to talk about some of the other issues and moving forward. So everyone knows, everyone has seen probably this slide in the bottom left, this diagram here about Moore's Law's ending. And so, how can we increase scientific accuracy when compute speed is slowing? And everyone here knows about all the exascale efforts and those things there. But, I think also perhaps based on some of the things I showed earlier about how the state of some of the biomedical software, how can we move that software forward and raise awareness and help really enable the biomedical community to take advantage of these resources and really move forward. So to me, this is part of a big driver for some of the things that we're doing. And I'm going to talk about that a little bit more, but I wanted to mention the reproducibility part here, because this is also an important part of this. So I was mentioning earlier that that many physicians, they don't check the smoking box, right? So then when researchers come and they get data from the data warehouse, or when they, how good is the data they're getting? They think that this is really looking at all smokers, even though it's not. Or even, you go into a doctor's office and, for the ethnicity thing, the people at the front desk may just be guessing, and how accurate is that? And then there's all of the, all the ETL processes, all the, from the IT perspective, all of those processes, and, what are we doing for error checking and making sure that as we, take the data from the medical records, put it in the database, do all these other things, what are we doing around that for error checking and all of these things? So I think that there's not a good understanding in the community about how there should be error bars on the data when we give it out, right? So researchers and clinicians understand what they're getting. And so I think trying to do some survey and have a better understanding of what others are doing and what the state of this is an important part of improving the accuracy and helping the research move forward in a productive way. So I think with some of these issues, these big issues, how are we going to move forward? So, some of these things I know you all here know very well, a lot of how can we have students or faculty, how can we make everyone at least conversant in computational and data science so that we can leverage either the existing tools, we have some awareness of performance, all of these things. So again, so we can get more science done, and a big thing has been being able to understand each other's language and have all these cross-disciplinary partnerships, because everyone speaks a different language and it takes a long time. People even use the same terms differently in different fields, and it's really I feel like it's taken me quite a long time to have and being embedded with all the different researchers and clinicians to really understand what the big issues are and what problems people are trying to solve and the big barriers, and then being able to try to leverage things from technology and other fields to try to marry with that so that we can move forward. So this is a big issue, and I think and so to me, this is where the training and helping to introduce computational thinking and everything, and infusing this not just into our curriculum, but down to K-12 and all of these other efforts in this area are so important. And so we've been doing some things at Sinai, but obviously this has to be infused throughout everyone, I would say. I don't know how I'm doing on time, but in any case, there's lots of ways. We've done various things to try to help at Sinai, but there's other things nationally. One thing that people may find interesting is we actually had this, so one of the ways we did this that I thought was kind of interesting, we actually had a decode your own genome course, which, I don't know, like if I could decode my own genome, I don't know if I'd want to, everyone has a different idea and you try to figure it out. But, we had this course and students could also pick a reference genome. They didn't have to do their own and the instructor didn't know if students were doing their own or a different one. So this was kind of, an interesting study, but, it was a nice driver that students were really engaged because they really wanted to understand how to do the computation and they really wanted to understand what the implications were. And some of the students taking this are in like the genetic counseling field. So they, when you do it on yourself, it's different when maybe you're giving information to someone else, but when you start to understand, how that would feel for yourself, it can change how someone may approach it for others. So this is a very interesting course and we still have it. Another effort that I thought might be of interest to some folks here is we have this FlexMed program. So this was actually started as a humanities program where basically when you're a sophomore in college, you can get early acceptance to medical school. So then you can major in English and you're already accepted. You don't have to take all these pre-med and biology courses at that time. And so now a couple years ago, we extended this for engineering and math and science. And by the way, the humanities people that we took, scored we have all the data showing that they did just as well in medical school as the people who weren't, didn't take all the pre-med and things. So now as, having the computer science, math, all this computational thinking is so critical to working on a lot of these problems that I was talking about in the beginning. We're specifically trying to attract these majors and give them the early acceptance to medical school. And we, are actively trying to get people like this. So this is super exciting because here you're doing, you're specifically attracting engineering, computer science, math, and around your new medical school that's here. So to me, this is very exciting and absolutely the right thing to be doing because we really need the different disciplines and people who can understand and band together on this is really important. Also, working with, also working with Verily, Google Life Sciences to have different internships and other things and infusing different capstone projects and other things in their curriculum. So students get exposed to different areas and different projects. I think this has been quite successful. There's a training grant that we got as part of the Big Data to Knowledge Award, and again, a summer school for computational genomics. We received also an award to do like an RNA school for spring for mid-career professionals. We have, so basically we pay for students to come to New York, or NIH pays, for students to come to New York City for 2 weeks, and we take them around, they go visit New York Genome Center, and we've done a bunch of big data trivia night, different things to help, again, pass on some of the computational thinking. And also we do these Dream Challenge teams, which perhaps you've heard of. I saw that Gustavo Stolovitsky gave a presentation here as part of this series a while ago. So he's one of the instigators of this. And again, this is bringing together multidisciplinary teams to work on different problems. We provided there was a mammography challenge just recently. We provided all of the de-identified data for that. And then we provide coaching. We provided some coaching as part of this award to again, to help people work together as teams in different disciplines, have some courses in Coursera, and we've done different BOFs and workshops at supercomputing and other places. We've also worked on partnerships with RPI and Stony Brook and Oak Ridge. Again, we don't have a computer science department, engineering department, so really, I'm seeking out other partners that we can work together to help work on a lot of these problems because there's plenty of them and we really want to make progress. So lots of exciting stuff, and working at this intersection of the different domains is really exciting and lots that we can be done. But, the main thing is the workforce and having people who can help we can bring to bear on these different problems. So That's all I have. Lots of people have helped me. I could not fit them all on the slide. So it's really, I've learned a ton, and it's been very exciting. So, oh yes, we have open positions. Come live in New York City. We have lots and lots of positions open, and yeah, come enjoy. So thank you very much, and thank you.
So we have a little time. It's a little over, but that's okay.
Sorry.
Is there any questions for Patricia out there? The young man in the front.
Hey, Patricia, thanks. That's
Hello.
great. I'm really excited. I have a couple questions, but I'll just maybe pick the first one about the difference between developing a drug where you have tons of regulations about it and developing an algorithm, and maybe there are fewer, or, you know, can you tell us a little bit about the regulatory side of developing algorithms and software?
I guess I would say it's difficult. I mean, it's interesting. There's different levels of, like, FDA has different levels, and you know, if you're good, like a home health device is not the same, you know, has a different level of regulation than if it's what you would use in a hospital. And I think, so I'm not an expert in this area, but there's definitely additional complexities and that go with that. So, I mean, so some of the things it's been interesting to do from the standpoint of, like, actually one of our, I'll just leave it at that.
Just like I could imagine, you don't just say, well, let me just change this prescription to like a different drug and see how it works. But you might do that with an algorithm, and I'm curious how flexible you are to sort of innovate around the algorithm space compared to the actual drug space.
Yeah, I would say we are flexible, but I mean, again, it's all these partnerships. So we have a bunch of partnerships with different pharmaceutical companies, and where we're able to do different experimentation. And like you said, I mean, there's actually an interesting article in the New York Times about all the efforts that have been put into all the molecular dynamic simulations looking for specific targets and everything, and what has been the outcome of all of those efforts. And I think, you know, it seems like in general the immunotherapy stuff has been a much more effective, has had much more, you know, it's like there were a couple good targets that were easy to find at the beginning, but it's unclear how effective that has been in general, I guess I would say. So we have, obviously, like you said, we can change the algorithms, but yeah, it's a different thing from the pharmaceutical companies and all the clinical trials and everything that they have to do. But we've had some patients that are, like, we had a patient who was like, give me any drug, I don't care. Like, I'm gonna die. So, you know, they had a certain kind of cancer. We sequenced a tumor. We did a drug, like we did a one-off thing. So there's been a lot of people that we've who've said, I don't care, I will take anything. I will be part of the experiment and like a one-of-me trial. And so that's kind of been interesting too, you know, so there's different things, but anyway, I don't know if that answers your question. But.
Thanks, Ed.
Hi, thank you. So I'm a graduate student and I do biomedical imaging, so MRI, and I've done a little bit at the local hospital with integrating, but I'm looking to do more, and it seems to be you agree with it, is getting people into studies is the hardest part. Where do you see the most success in getting them integrated? Because once we get them in, they tend to be really happy to help, and they end up being actually better subjects than healthy people because they're used to being poked and prodded and everything else. So it seems like they're eager, but they're not getting into the funnel. So how do you, what are your recommendations for getting more people in?
Yeah, I mean, this is a whole area of interest. I mean, I think some of it is, like, you can go to clinicaltrials.gov and search for clinical trials, right? But most people don't do that. And I think a lot of the thinking is that people just don't know. So one of the things is we're trying to have alerts and epics. So when a physician meets with a patient that, oh, this person's a candidate for this trial. So we're doing things like that. But yeah, so what's the answers? I mean, I think that's where like some of these apps, like I want to participate in the asthma, like the asthma app was around an asthma trial, right? So some of this is reaching people in different ways, or going to like this Patients Like Me and some of these other groups and trying to find other ways to get patients. I just worked on an atypical diabetes proposal where this was a big part of it, was trying to, how are we going to reach these communities? People probably don't even know they have atypical diabetes. How do you find these people and recruit them and convince them? And so for this project, it wasn't just finding them and convincing them. You'd have to get, I think it was 4 other members of their family so that we could look at compare the genomes and see the patterns of, all of these, hundreds of people. And so how do you convince all these people to do that? It gets tricky. So I don't know that we have all the answers, but I think, it's like different kinds of engagement. Some of it's like, oh, maybe we need a Facebook on this. Some of it I think is the social media, and some of it's maybe patients can appeal to people directly. But yeah, we do a lot of like searching through the medical records and looking for patients. Another thing we have is there's this company called TriNetX that like we're sharing de-identified data with them, and they have like 81 million patients. They've worked with like 25 other medical centers who are sharing de-identified data with them. And so you can search on 81 patients. TriNetX is a spin out of a pharmaceutical company, so you can search on 81 million patients looking for specifically for the patients you're looking for. But again, like, helping to recruit the people actually into the trials is different, even if you identify them. And that's, again, based on the data that, you have to think about what's the quality of the data that you're looking for these patients on. But yeah, you can identify. So yeah, it's a big problem. That's the short story, and those are some of the things we're doing.
So related question, how do you get people to consent to have their data in a data set so you can analyze it?
So that's a really good question. Let's see. Some of it is, so there's different models for consenting. Like some places there's some opt-in models, there's some opt-out models. Like for our biobank, all those people, when they opt into it, they say they're okay to be recontacted for future trials. There's a whole bunch of different models around this. So there's a whole bunch of different models for doing this, and different places have done different things, and I think it depends on what you're doing. But perhaps maybe what you're really asking me about is like the data warehouse. So every patient that comes to Mount Sinai, they can be used for internal research studies that are approved by the IRB, that's for the protection of human subjects research. And so, when you go to the doctor and you're signing all those forms that you maybe don't read, I mean, that's part of the consent to say that your information can be reused in that way. So, and I think a lot of medical centers are trying to, have different opinions about sharing their data, with outside institutions and other things. So, I don't know. I suggest everyone reads all of those documents they give you, if you don't want to be included. So there's a whole area around all the consenting stuff. So I don't know if that answers your question, but.
I was thinking in the light of Equifax and everything like that, people are going to say, oh, no, I don't want anybody having my data.
Exactly. Yep.
Any other questions? Well, let's thank Patricia.
Okay, thank you.
Thanks for being in school.
Oh, no.